In this video, we will discuss about a new paper where we will uh, understand how this DiaBZl helps in increasing the T cells cytotoxicity against the tumor cells by enhancing sting that will increase the antigens that can be detected by the engineered T cells. So, how they have studied this topic, we will go through each of their figure one by one. So in the first figure, the panel A, what they have studied is that they co-culture these effector cells, the T cells in 1:4. And effector cells are the engineered T cells, and the T target cells, the target cells are the tumor cells. So in the first case, the ratio between effector cells and the target cells is 1:4, and they use this DiaBZl compound to increase the cytotoxicity of the T cells against the tumor cells. And how the DiaBZl works is DiaBZl will increase the sting in the tumor cells. That will increase the amount of antigens that can be detected by the engineered T cells. We will discuss about the engineered T cells in the panel D. So first, what they present in the panel A is that the results of the cytotoxicity when the effector cells and T cells are treated at 1:4 with DiaBZl. So we can see that when the DiaBZls are used at 1 microgram, they can see the increase in the cytotoxicity. However, when the DiaBZl is increased to 10 microgram, the cytotoxicity goes down. And one more thing that they have observed is that when the increase the number of effector cells, the cytotoxicity increases both in all the three cases. However, the maximum remains when the DiaBZl is used at 1 microgram. And when in the panel C also we can see that when the effector cells are increased further, that is around 1:1 to the tumor cells, the cytotoxicity goes up compared to the other two cases. However, the trends remain the same. That is, at 1 microgram, the cytotoxicity is maximum. Then for this 0.5 microgram, but it decreases for this 10 microgram. And they have tested it on the three different cells. First one is this MEL526 T cells, and the combined, not three different cells at three different conditions. We can say that. First one is this their cancer cells line, that is ME1526 and the T cells, and the third one is the co-culture of this ME1526 and the T cells. So what they have find out is that when they co-culture them, the cytotoxicity goes down when they increase the DiaBZl to 10 microgram. However, for the 1 microgram, the cytotoxicity is higher. And they can also see the cytotoxicity of the DiaBZl on the T cells also. That is, T cells also cytotoxicity against the T cells also increases with increase in DiaBZl. So it should be used in a minimum amount. It should be used in a optimum amount, not minimum amount. So in this panel C, it looks like the 1 microgram gives the optimum concentrations that can be used further. So in the panel D, now they have discussed about the lentivirus transduction of these T cell receptors. So they have used these two, that is IgA4 high affinity T cell receptors and IG4 wild type T cell receptors. These high affinity T cell receptors has more cytotoxicity, and it is found, and it is also used in some of the clinical trials also. And this is the wild type IG4 receptors that is integrated into the T cells, and the quantifications of this lentivirus transcription is shown in this panel E, or lentivirus transduction is shown in this panel E, where we can see that the transductions can be seen to this T cell receptors to about 80%, and also about this slightly more for this IgA wild type. So once they have the engineered T cell receptors, they wanted to check whether there's antigens that can be detected by these receptors, that is IG4HA and IG4 wild type, are expressed on the tumor cells or not. So one of those antigens is this NY-ESO1. When they checked in this MB231 and ME1526 cells, they can see that both the antigens are present. However, in the case of MB231, the antigens presence is higher compared to the ME1526. So since the antigen present is MB231 is higher, they are expecting more cytotoxicity on this MB231 compared to the ME1526. And when they check their results, they found the similar result. They found the expected results, that is for this ME1526, since the antigen expressed on the this ME1526 are lower, they see the lower cytotoxicity compared to the case of this MB231, which has higher antigen expressions on the cell surface. So they see the higher cell cytotoxicity when they are treated with the effector cells or the engineered T cells. So from this, their hypothesis seems to be plausible, that these will cause DiaBZl will cause increase in the antigen expressions on the tumor cells, and those increased antigens can be detected by the engineered uh, T cells, that is T cells engineered with these receptors Ig4HA or Ig4 wild type. Ig4HA is Ig4 high affinity T cell receptors, and Ig4 wild type is the Ig4 wild type receptors integrated into the T cells surface. So I hope you, and this is the another these two are the controls. So I hope you understood uh, how they have provided their hypothesis, or how they have, from this figure 1, they have made a point that their hypothesis seems plausible. If you need to understand more about this topic in detail, or these experimental results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.